Thank you, Victor, for the introduction, and thank you everyone and the organizer for providing this opportunity to present this work and the more general work that will be further introduced by Anne. So can you see my screen, Steve? Yes?
Yes, yes.
Yep. All right. So I will introduce the work that been published last year that we did here in the lab and in collaboration by different teams that focused on the benchmarking of T-cell receptor repertoires profiling methods and showed some bias between the methods. So just before, although this the people that are attending this meeting, I'm sure they are all aware of this, but just as a very brief introduction, you all know that B cells and T cells express antigen-specific receptors. This is how they are called, and we name them TCR and BCR. Each of those cells express, in fact, each B cells or each T cell express a unique TCR or unique BCR, which is generated by random somatic recombination. And they are all clonally selected based on those TCR and BCR, which leads to a collection of clonal T and B cells that is what we formed the TCR and BCR repertoire. In terms of numbers, why we are interested in comparing some methods, one important thing in the field of TCR, and it's the same for BCR, but from now on, I'm going to focus on the TCR, is that the number of unique TCRs that are generated are very huge. So the potential is up to 10 to the 19th TCR that can be generated in the thymus. And then after different selection process, you can still find in one person's body at a given time from 10 to the 6, 10 to the 8 unique TCRs, which makes a huge diversity of molecular species and proteins that can provide an immune response. And this collection of unique TCRs can be modified during any immune response in the presence of infection, tumor, tissue damage, or whatsoever. So for which we usually don't know the diversity nor the modification. And this is exactly what people like to analyze when they are studying the TCR repertoire as a way to understand the physiopathology and the modification of the capacity of the immune system to respond in this context. So those numbers show that there is a huge and dynamic diversity of TCRs that can be analyzed across time and space, because this can be also different between the tissues and the place you are analyzing. So how those repertoires are analyzed. So this is kind of a historical retrospective, but I'm going to be very fast. So for the repertoire in general, especially the TCR, we started using flow cytometry, and in the late 90s, there were these methods of using immunoscope or CDR3 spectrotyping that already provided some information in terms of diversity. A major lack of this method was that we could not get direct information about the sequence, and then the clonality of the rearrangement was not easily assessed. So in the 21st century, next generation was deployed for genomics in general and transcriptomic, and progressively was applied to the study of TCR and BCR, which we call now the adaptive immune receptor repertoire sequencing method. So with these different methods, we can definitely understand that the data complexity that is generated based on the diversity of the TCR repertoire is highly increasing. So when we are analyzing the repertoire, we usually we are generally interested in looking at some particular features, the one that we can retrieve from the data. So here it's a general picture. I'm not going to enter into the details, but of course you can analyze the samples one by one, or you can compare the samples between themselves. And when you do the analysis, you can analyze the usage of the different V genes, J genes, combination of V and J, for instance. You can also get the CDR3 length distribution, the frequency of the clones within each CDR3 length distribution or overall. You can compare the similarity in terms of repertoire between different samples to see how much the clonotypes that are present in one sample to another are close enough or quite different. And you can also do some gene expression. There are more sophisticated and advanced analyses that can be done also currently, and that, for instance, focus on the network analysis and similarity between the different clonotypes within either one repertoire or between repertoire, and also convergent selection. So because we are looking at different parameters at different levels of granularity, there is a need to really have good methods. And when you consider AIR sequencing using next generation sequencing, and especially here I'm going to focus on bulk sequencing, there is several steps, several levels that needs to be considered to have interpretable results and good data. So first, the sample type, then the data production, and of course, then all the methods and tools for analysis and integration. And all these four steps are related to different challenges that are tackled by many teams around the world. And the one I would like to focus today are the one regarding sample preparation, the sample type, and the data production. Why? Because sample type, when you are analyzing the repertoire, there is many ways to do it. You can start from different tissues, organ, or from the blood. You can have access to fresh or frozen, sorted T cells, whole T cells, or PBMCs. So it depends on what you have access and what is your question, of course. There's a possibility to analyze the repertoire either from DNA or RNA. You can have access to different amounts of this material depending on your setup and depending on what you are looking. And regarding the data production, there's many methods that have been developed to try to amplify those TCRs to be sequenced afterwards. So in this presentation, I will mainly focus on these two main categories. First, the type of nucleotides that are analyzed, either DNA or RNA. The input quantity also will be discussed at the end of the presentation. And the main point will be the comparison of different methods with a focus on multiplex and race PCR, which are the most used ones. So in terms of methods, as I said, multiplex and race are the most used methods. They have been developed either by academic labs or by different companies, and those are the leading companies currently for those methods and for which we have the more data. You can see that depending on the methods, the template nature is either or both RNA and gDNA, and they use different protocols. So the question is whether all those methods can generate the same type of information and the same comparable repertoires when you start from one sample. So before our study, there were already some studies assessing this point, and those are the two ones. Those studies had the possibility, well, they analyzed 2 or 3 samples from 2 or 3 different individuals, which was a very good point. But usually for each sample, they had only duplicates, which in terms of reproducibility and replicability, it's hard to evaluate. In this study, they use cells from frozen tissues, here total PBMCs. They compare 2 to 3 different protocols, either comparing RNA and DNA or just RNA, looking at either TRA and TRB rearrangement or only TRB. And the important point also that will be further discussed after by Anne is that in the first study, they had no controls, internal controls for the methods assessment. And in the second one, contrary, they had different controls, but assessed on DNA instead of RNA, and that was the input of their use for their methods comparison. So we decided in collaboration with international or with other researchers working in the field who developed themselves different protocols for assessing the TCR diversity by next generation sequencing and using different TCR methods to compare these methods. The idea was to select those methods that were the most used, so either developed by academics or by companies like Adaptive, and in our case, we compared also iRepertoire methods. And it was also voluntary based. The idea was to challenge that each of us would challenge the methods we were using. So the general design of the experiment is the following. We started from one samples of CD4 plus CD25 negative sorted cells, T cells. And we did this because this is what we are interested in the lab, and we believe that it's important to assess the repertoire by looking at least T cells and a subset of T cells. So we had a sample of T effector cells for which we could extract both DNA and RNA using a trisol extraction. And we also prepared a DURCAT clone. So the DURCAT clone was used as a control, internal control, since this is a cell line with a unique TCR alpha and a unique TCR beta chain. And we decided to use this at different concentration which is shown here by the dilution, so that to be able to see whether this particular clone could be detected, whatever the concentration, in a linear manner. So we prepared for each method 3 aliquots with 3 dilutions, which led to 9 aliquots per method. And this is the list of the different methods that we've been comparing. 2 of them were based on DNA, and the remaining one on RNA. And as you can see, there is definitely a shift between multiplex and race, since race PCR has been much more used and developed by academics and in one company. So the first thing I wanted to emphasize is this, the linearity of the clonal spiking that we assessed. So here you can see for the alpha chain and the beta chain of the DURCAT clone, for each method, one after the other, what was the frequency of the TRB or the TRA that was detected by sequencing compared to, so that is in blue with the different blue points, compared to the theory, what we would expect to have in our sample. So first of all, regardless of the methods, we could detect both all the dilution, the TR alpha and beta at all the dilutions. Interestingly, we could see that the frequencies of the TCR alpha is 3 times lower than expected, which may have some consequences and could have some interpretation in terms of the transcript of the alpha chains and the stability of those transcripts, at least in this GERCAT cell line. For the TCR beta, it was the opposite. We had 3 times higher, except for the multiplex PCR1. And overall, you can see that the linearity is maintained, meaning that we can see the expected fold change regardless of the method. So that was for the general assessment of the methods, but then we wanted to go more in details and to see whether all the methods could perform similarly in terms of all the parameters that we are usually assessing by analyzing the repertoire. So we looked at the usage of the TCR beta chain and compared whether those usage were similar between the methods, the different methods, and what could be expected from flow cytometry. So of course, here there is a gap because flow cytometry is detecting the protein, and we are analyzing either RNA or DNA. So in this chart, you can see the frequency, the ordered frequency of the 24 V betas that could be stained with the kit we used. And when we looked at the frequency of those same V betas with the different methods, the first striking point is that for all the RNA-based methods, there is some V betas that are almost not detected by flow cytometry that are highly expressed or seen using RNA. And the second important point is that one of the genomic DNA-based methods is fully linear compared to flow cytometry. And this can be seen here with the correlation analysis. As you can see here, this MPCR1 is the most correlated. However, when you compare, when you analyze the correlation between all the RNA-based methods and the other methods compared to the flow cytometry, you can see that there is several methods here, especially race 3, 4, and 2, that performs quite well in regards to what is expected in terms of cell population at the protein level. So we further analyze and dig into this gene usage, and for this we analyzed all the parameters that can be analyzed from the gene segment, meaning the usage of the V gene, the J gene, the insertion between V and J for alpha and BD or DJ for the beta chain, the deletion also at the end of the V or at the beginning of the J. And we analyzed first what we called the repeatability, meaning the similarity among the data within the same methods, to determine whether for each method all the technical replicates were similar or not. And in a second hand, the reproducibility, which is the similarity between different methods. Altogether, what we can say is that depending on the chain that you are analyzing, the methods will perform differently. So for the TCR alpha chain, we could see that there is at least 2 methods, race 3 and 5, that are the most replicable, here in blue and green, and the most reproducible. Other methods are not that much replicable, meaning that when you are doing several replicates of the same sample, you may not get exactly the same results, while they seem to be quite reproducible across the different methods, meaning that somehow they can detect overall the same information. For the TCR beta, there is one method which is replicable and reproducible, which is race 3. The other are overall rather reproducible, but not that much replicable. And for instance, this is the case for MPCR1 is less reproducible than race 3, for instance. And one of the methods that was finally excluded from the study because it was really messy and in fact not developed for the repertoire analysis is the race 6 that you can see is neither reproducible, replicable, nor reproducible. So the first take-home message here is that already when looking at the high granularity of the repertoire, depending on the methods you use, you may have some impact in terms of reproducibility and replicability. We then compared the similarity in terms of clonotype sharing. So for that, we use Morrisita Horn index or the JACAR index that can assess how much two repertoires and two lists of clonotypes are close enough in terms of present substance and the frequency of each of them. And we did this for the TRA and the TRB. As you can see again, there is some methods that are quite highly similar. When they are red, it's Morrisita Horn 1, means they are similar. So some methods are highly similar between themselves and between others. For instance, here for TRA, race 3 and 5. For TRB, we have more methods that are similar, still with this race 3 being the most similar between within itself and between others. And another important point is that multiplex PCR seems to be completely out of comparison with the race PCR. And this is not only due because here Morrisita Horn take the frequency, and when we do multiplex PCR on genomic DNA, we can say that we can think that the frequency of the clonotypes are different because genomic DNA and RNA are not assessing the same. But in fact, when we do JACAR analysis, which is not taking into account the frequency, we got the same result. So there is definitely a gap in terms of the list of clonotypes that can be obtained between the different methods. So to further assess the sensitivity and the replicability, we made a meta repertoire, and this was done so considering that at the end, for all these methods, we could get some list of clonotypes, more or less sharing, and we could see whether each method could detect something out of the other method. So we had 9 data sets per method. We pooled them. We removed the singletons to remove noise. And then we remove all the private clonotypes that were seen only in less than 3 methods, so that we could have we could remove the noise due to eventually a PCR or a sequencing error. So that leads to smallest data that were further downsampled so that we can get similar number of clonotypes for all the methods, and we pulled everything, and this is what we called a control meta repertoire. So then we use this control meta repertoire to assess each method again. And one thing that we first tested was how much of this control repertoire clonotypes we could find in the different methods and considering the different replicates. So the dot here, for instance, shows that there is about 20% of the clonotypes that is present in the control meta repertoire that have never been detected with race 1, and 40% that have been observed in one replicate of race 1, 20% in 2 replicates, and so on and so forth. So the general idea of this figure and this result is that we have most of the methods, as you can see here, that are not detecting about 20 to 40% of the repertoires that can be detected overall across the different replicates and the methods, except race 3 and race 5 for TCR alpha and race 3 for TCR beta that seems to perform a bit, well, much better, including being able to detect more than 10% of the control repertoire out of the 9 replicates in this method. And finally, another important point was to determine how sensitive are the methods in detecting rare clonotypes. And this was a particular question of interest for us because we are mainly studying autoimmune diseases and probably rare events that would be involved in the disease. So here I'm showing the frequency of the clonotypes that are present in the control repertoire that are either not seen at all in any replicates of the different methods, or seen in the 9 replicates of each method. The frequency shows the lower is the frequency, the rarest is the clone. And here again, you can see that there is 3 methods that outperform, raise 3 and 5 for array, for TCR alpha, and raise 3 and multiplex TCR 1 for TCR beta, that were able to identify rare clonotypes in 9 replicates after 9 replicates of the same sample. So, and before finishing, in the last part of the study, we wanted to evaluate also the impact of the RNA quantity, because this is also something important for us. Depending on the sample you have access to, you may start from very few material or much more. And we wanted to see how much those different methods were impacted by this variation and what was the impact of the variation in quantity. So with no surprise, the less material you put here in green, 10 nanograms, or in black, 100 nanograms, the lowest is the richness, which was expectable. Interestingly, when you look at the diversity of the most abundant clonotype here in the RNA profile, you can see that regardless of the input quantity, the proportion of highly expanded clonotypes are maintained. So this is a good point because it's probably something that we might be interested in. And the last part was the comparison of the V usage or the clonotypes, again, using Morishita horn to see how much our replicates were similar. So we compared replicates with this made from the same quantity, here 10 versus 10, 100 versus 100, and also 100 versus 10. And again, you can see that depending on the methods, the Morishita horn index, whatever the parameter you are analyzing, is variable. Especially looking at the clonotypes, most of the methods have a very low Morishita horn, especially for low amount, except eventually raise 3 that has the highest. And on contrary, when you compare 10 versus 100, everything is a bit increased, but still raise 3 is the most abundant. This is true for alpha, the alpha chain, but also for the beta chain. So this, out of this study, we could assess the different methods, 7 methods based on race or multiplex PCR, and based on the results we obtain in the different analysis, we attribute a score. The lower is the score, the better is the method based on the analysis we made. And this led us to choose one method, one technology, one technique for our own study, which is the race-3, as it was the one with the lowest score in terms of replicability, reliability, and sensitivity. In terms of cost, was not very costly compared to other methods, which is also important when you have many samples to analyze. It's provided through a kit, quite easy to use. So again, this is important in terms of reproducibility in the lab and sharing between students and technician. And of course, you have access to the data since you are the one producing them. So to conclude, so from this study, what we saw is that depending on the methods, the performance performance are different between and within methods, and this based on different criteria and parameters that we've been analyzing. And we believe that having this knowledge is important for data and results comparison. I would like to stress that knowing this, it's really important to follow the AIR standards recommendation from the AIR community, which is asking to get the information about which methods and which sequencer and so on and so forth have been used for the generation of the data. That could be of interest for further analysis, and maybe the data set we generated in this study could be of interest also to evaluate tools, new analytical pipelines, maybe developing some correction tools to put all the data in line. I don't know, maybe this is a utopia, but and the last point, which is more perspective, and Anne is going to talk about it more about this, is that we need some internal control, because here we made a huge comparison, so that's fine. We found our methods, but there is always the risk for any issue to happen during the experiment, and having those internal controls for every daily experiment could be of interest. So you can see more details in this review and after the discussion with the presentation. Thank you very much.
Thanks so much, Encarnita. So I think we move on to Anne, and then we have the question session together for the both of you.
Mm-hmm.
Okay, can you see my screen?
Perfectly, yes.
Okay. So good afternoon, everybody. So as Encarnita already announced, I will talk about controls and standards, and I have to disappoint you with Victor said I would present something on BCR, but it's not something, it's no results, it's a prospective, so a planned, something on what you plan to do. So actually I say I'm speaking on behalf of the AIR Community Biological Resource Working Group, because what I will present now is actually the result of the now years-long very fruitful discussions and what we have done together to design what we want to do in terms of standards and controls. So when we started in 2017 at the AIR community meeting, we got together and we said that we made our goal to provide the AIR community with standards and controls to enable the researchers to work in a harmonized and standardized fashion. So that was our goal. And we decided to start with two things. So we decided to do first a survey to really see and get into what people need and what there is around. And secondly, then to review what there is around in the literature and what standards are already used by writing a review. So the survey first, was up for 6 months, and it was distributed in the whole community as far as we could go, also through IMTG. And we received 100 responses that were usable. It contained 4 sections, and there were cross-check answers too, and also open answers to go into. The survey was then published in this eLife review that we published in May, and the results were there. And I will quickly just show you what we obtained. So first of all, we wanted to know where people came from and what people did. So 96% of the participants were already involved in AIR sequencing studies. And you can see at the left top that most of the users or the participants were based in Northern America or in Central Europe, but there were also users in Canada, Southern America, in Russia, and also in Asia. So it was a bit all over the world. And then we wanted to know what these participants were doing and were they interested. And most of them, as you can see, were working with human samples, but there was also some mouse and some other animals that were studied. Then the distribution between BCR and TCR, you can see that most users were interested in IG, so in BCR chains, So it's almost half of the users, but some were also, a quarter was using looking at TCR, and some of them also were looking at both, so interested in both. Then what did people want to study? So here in the center, you can see the big pie chart, and that shows that most of the people were very interested in immune system diseases as infection, autoimmune diseases, cancer, but mostly infection. This was the main thing. And then there were also people that were using AIR studies to develop a bioinformatics tool, to look at vaccine responses, immune repertoire, and so on and so forth. And there was also a section with other, which comprised hematology, aging, and some people were also just doing small things. So this was basically the overview, was what people were doing. Sorry, we went further and then asked what methods were used. As you have seen before, as Encarnita presented, it's very important to see, to decide what method to use, and therefore we wanted to know what was around and what people were actually doing at the moment. So mainly people were using bulk sequencing. So 91% of the people were doing bulk sequencing, 8% at the time single cell, and I think that now probably has changed a lot since the development of 10x genomics, and 67% were doing both. And as you can see here, there was also a plethora of methods used. So there was multiplex PCR and template switching. That was well explained by Encarnita, was the majority of the methods that were used. And then there was also some target capture and total RNA-seq by some users. And some users were also using different methods, but most of the users were just using one method. So either multiplex or template switching, or then both. We then also looked at what the starting material would be. So as you can see here, 2 blocks, either using multiplex PCR or template switching. And in both blocks, you can see that the majority of the users were starting with RNA, which is in green. For multiplex, we had then a number of people who were using DNA, and some were using both. And in template switching, it was mostly RNA, but also there were also some people that were also using both. And then we also looked at UMI, non-UMI users, and we see that the majority was not using UMI for multiplex, and if using template switching, of course, there were more UMI users. So summarizing this, RNA was the preferred starting material. We then wanted to know whether there were participants that already used controls, and of course also to learn what these controls would be, to be able then to design our own controls. Actually, the 47 of the participants, so almost half of them, already use controls, which was interesting. And then you can see 23 of those, so almost half of those, were using homebrewed controls, so things they designed themselves, whereas the rest was probably using some commercial things. And the favorite controls were cell line for RNAs or pool cell line preparations, but there were also cloned plasmid, DNA fragments, in vitro transcribed RNA, and then other things like DNA quality assessment assays or cell line for DNA. And then, of course, the last part was asking the people whether they would be interested in using controls in the future and what these controls should be able to tell them. So, of course, most of the people were interested, so you can see here, and you can see in gray the ones that were already using controls, and in black the ones that not yet, and then that would like to use controls. And you see that most of the people were really interested in controlling for sequencing errors, followed by sensitivity and detection of contamination. But then there came also the calibration, sample quality control, evaluate also the batch effects, specificity, linearity, and clonal expansions. So this has led us, summarizing this survey led us to really say there is a need for controls and standards in the community. We really need to develop something. And this is now the second part. What I will tell you, this is now a perspective of what we want to do, what we are starting to do now at the moment. with the Biological Resource Working Group, we have plans to develop standards, and to do this, we want to do a B-cell receptor benchmarking study that resembles a bit what Encarnita had done. So we will focus on BCR because most of the controls that were already available in the community come from BCR, and also because most of the people use BCR, as you've seen in the survey. We will focus on IgH because it's the most diverse chain. First of all, I want to also just raise your attention that what are the differences between the TCR and the BCR? So we have to take into account that the BCRs are more complex and more difficult to analyze because of the somatic hypermutations that we have. And also we have several isotopes, which we don't have in the TCR. So this makes the whole story a bit more complicated. Then what do we need? What do we need to control? So this recapitulates what we have seen with what Encarnita said, and also what we have seen before through the questionnaire. So we want to control sequencing errors, sensitivity, contamination, specificity, sample quality, batch effects, clonotype quantification, and also reproducibility. So our plan is to generate samples for standardized BCR repertoire analysis, so biological samples that we can then, in the end, distribute to the community. We want to design the samples, produce them, and test them on two methods, with using gDNA, genomic DNA, or RNA. And to do this, so this first step, we have now submitted a grant application written by Nina and by Andrew Farmer from Takara. And this has been submitted to TAPS, to the TAPS and AIRR-seq initiative. And we hope to get the funding to do this first step. And in a second step, we want then to use the standards to do a comparative methods over several centers, similar to what Encarnita did for the BCR repertoire sequencing. So a multi-center study. First, so the first step, so to generate the standards. So our plan is to use three types of samples. First, the first type would be a polyclonal mixture of spleen that comes from a donor, from an organ donor. And the advantage of this sample is that it has been heavily used already by the lab of Nina, by the Munich Prague lab. They have used this as a kind of a run control for all the sequencing they did, and they have now a plethora of data at hand that can then be compared to the results that we will get afterwards. This mixture of spleen contains a mixture of mutated and unmutated clones. So we will start from this spleen, we will obtain genomic DNA or RNA, and we will start with two sample sizes, 100 nanogram and 110 nanograms as an input. The second plan we have is to use polyclonal cell mixture. The beauty of that is that we can access different isotypes. So we will try to find cell lines that cover all the isotypes available. We will mix those cell lines and we will extract gDNA or RNA, and we do a titration of those cell lines into PBMCs with a 10-fold dilution series that can then be used as a standard. And the last control will be a mixture of synthetic fragments. And this has been already used and tested by Johannes Truck. And you can see here, this mixture contains a number of combinations of all V genes and some of the constant regions, as you can see here in different combinations. And all in all, we obtain a large number of different possibilities. And those fragments are in vitro produced, and they are all the same size. So they have 511 nucleotides. They contain a random CDR3 region with a tag in the middle, and they give the possibility to use a multiplex V-gene PCR primer site or a singleplex PCR with a primer site that is added at the end. Those synthetic fragments the mixture of those fragments can then be added at different molecule numbers, and then be very well controlled in this way. So this, to summarize this, we will use those three types of samples, and we will use two methods that we will test. So we chose to go for methods that we have actually described in a series of book chapters that has been submitted to Methods in Molecular Biology, and where our group, the bioresources group, was heavily involved in writing the lab chapters. And it will hopefully be published soon. So two of the methods were described there, and one is the homebrew method from the Luning Prac Lab from Nina, which is already published as well. And this is going to focus on genomic DNA and will be done in the Luning Prac Lab. And the second method is based on RNA. It's actually the smarter human BCR kit from Takara. And Takara has offered to run those experiments at Takara itself, and Andrew Farmer is leading this. So we plan to have a number of samples. So we'll have the spleen mixture. We will run duplicate for RNA and for gDNA. The polyclonal cell mixture we will run for each dilution series for RNA and for gDNA, so make 6 samples. And then we have the mixture of synthetic fragments, where we will have the fragments alone, fragments in PBMC and fragments in spleen, and every time in duplicates. So this is what we will run. And so just to go through the parameters that we want to control, to repeat this. So we will have, sorry, just a second. We will want to control for sequencing errors, which can come, of course, from enzymatic reactions at the level of the RT or the qPCR, but also from the sequencing itself. And this will expect to use, and we want to use cell lines or the synthetic controls. We want to retrieve the expected sequences and then use this to evaluate the error rates. Then we want to detect contamination, which was also an important requirement from the community. For this, it's easy. We will have a unique spike in for each sample, and so we can then see whether we find contaminations of the spike-ins in each samples. Contamination, of course, can occur at each step, and it's mainly due to the bench work. Then we want to control sensitivity, and this is again, sensitivity can be influenced by, first of all, by the sample that we obtain, by the number of cells, the fitness of the cells, the state of the cells, but also again by the processing of the samples and by the enzymatic reactions. Therefore, we plan to use the spleen sample, where we can see, look at the number of clones that we found in repeats when we compare the repeats. And also we can compare this, the data we obtained with the data that has already been obtained by the previous sequencing experiments by Nina's lab. The cell lines, of course, will allow us to test for presence and frequency and linearity of the titrated spike-ins, and also the controls and synthetic controls in spleen, where we can look at the effect of spike-in on sensitivity when we have the spleen alone or the spleen with the spike-ins. Look at specificity or VH bias, which of course can occur, what is a danger when you use multiplex PCR. We can again use the spleen sample, where we actually do also expect to find a bias when we compare RNA to genomic data, because of course the RNA based method is variable due to different transcript number, activation, and so on and so forth of the cells. And we can also control the somatic hypermutation effect on primer binding on the genomic DNA. And this can be looked at the spleen sample. Then also we expect to find a bias maybe in recovering mutant templates. And there we can compare frequencies of muted and unmutated templates. The cell line will help us to compare the bias in isotype amplification. There are RNA methods, and the same with the synthetic fragments will also help us to distinguish this. Next, we also want to control clonal size distribution or skewing. And there, the spleen sample will be very helpful, where we will compare 20 clonotypes, the 20 top clonotypes distribution between the two sizes of sample input and between replicates. And we will be able to see what the impact of low input is doing to skewing. Then the replicability and reproducibility. Of course, this can be due to factors during the whole process and at the bench, but also at the analysis part. And there we will just simply compare replicates of the same sample at one center. And later we can then also to control reproducibility, we can then also control replicates between samples at different centers, but that will be in the latest, in the further step. Quality control can be controlled by using current methods, spectroscopy or gel electrophoresis, which will be done in the labs. And then finally, batch effects, which is again due to preparation problems or also in the sequencing. This we can again control with the cell lines and the synthetic controls that we use and compare samples and batches, and also compare parallel spleen samples in batches. Then data analysis, this will be done in the labs, so that in the Munich Prac lab and at Takara with their own methods first, and also with the common methods. And this common method still needs to be selected. And so there we would be really happy if some people from other AIR community working groups or the AIR, for example, the AIR software working group would collaborate with us and would have ideas and propositions to give us. And finally, the data will of course be, as far as possible, deposited into iReceptor so that it will be available for further studies. Then the second step, as I mentioned, I will just quickly say what we plan to do. It will be very similar to what Encarnita has been doing with TCR. It will be a multi-center studies. The methods that we will be using need to be discussed, but we are very open for suggestions. So we will start with RNA or gDNA. And of course, we'll use the Munich Prac method that is established, Takara method. But if some of some people from the community would like again to challenge their own methods or test their own methods and have other good methods to test, so we're very happy to take suggestions. The groups to be involved will depend on first of all on the funding that we will obtain, maybe, or also on people that how many people will offer their methods to be tested. The materials that we will use, of course, will be the standard samples that I just described, and the sequencing we plan to do centrally to avoid center-specific effects. And then again, the analysis tools, this we hope to be able to select together with the software group as to find the best possible methods. And with this, I would like to thank first of all to the Biological Research Working Group, which is really a very productive discussions we have had over this now 3 or 4 years. And then, of course, especially to Nina and Andrew Farmer, who have written this grant that we have submitted, and to Johannes Truck for providing the synthetic fragments. And now I think we are ready for questions.
Yes, thanks so much for these 2 amazing talks. And if you have questions, please write them either in the chat or unmute yourself, or raise the hand and then we can give the word to you. So I can maybe start with one question to Anne, which is based also on the work by Encarnita's Nature Biotech paper. So now that we know that there is so much variation, or that there is some variation across protocols, and that we still have the problem with normalizing across protocols to actually merge the data sets, how will those controls that you now design, those bikins, really help us also correct the entire data set? This is not clear to me. Boaz, can you please mute your? Yeah. please go ahead and answer. Yeah.
So you mean to correct the entire data set?
Well, yeah, so you have your spike-ins, right? And then you can compare them across the labs and across the different techniques and so on. That's all fine. But what we want to correct is the entire repertoire data set, not just knowing about the spike-ins, right? So how will the spike-ins help us with actually normalizing repertoire data sets, not just the 100 spike-ins that we spike in, basically. Yeah.
But I guess, I mean, from the spleen sample, we have already a complexity that is resembling a real data set, I mean, a real repertoire. And we hope that by finding the right methods with the help of course, the bioinformaticians, we will be able to prepare something that we can then use in a standard manner on real data sets. I mean, that's the way I imagine. I don't know whether it's feasible, but
it's not really what I mean. So you have spike-ins, right? So you have them in your data set, but how will it help you to correct the non-spiked-in data? That's not clear to me.
So I think for instance, spiking cell lines or even the standards, the major point would be first to assess the clonality, the quantity, the frequencies. So if you see any major difference across all spiking, because I think this would be the most important to see whether, depending on the spiking, you see the same differences or not, which is not clear yet. Eventually, you could assess a correction in terms of frequency overall, but which is yeah, absolutely, especially for for all the spiking. One thing that can be easily done, I think, is already to assess for the error sequencing errors. So that could be helpful to maybe modify the tools for analysis to be a bit more stringent or a bit less stringent if at the end you realize that there is not so many errors and in fact we are removing too much information. Then in terms of normalization between sample to sample, this is a really hard question because actually you don't know what you're looking at.
Yeah.
So I think it's more for at least quality. So just to ensure the first, right, to ensure that what you have, the data that you have are reliable and they are not suffering from any strange problem that you could have during the experimental setup, the wet part.
Okay, so we are running a bit over time, so maybe just 2 questions from the chat. So Felix is asking, I guess, to Encarnita, why was race 3 and 5 better? Yeah.
So why? I don't know. Actually, race 3 and race 5, and most of the race use most of the race protocols use almost the same regions, especially race 3, race 2, and race 5. I think the enzyme were the same, so it cannot be attributed to such kind of things. Now it's probably a matter of primers, maybe the primers that are used for the template switching. As Ivan said, maybe the template switching quality, oligonucleotides that are a bit different, and that we did not assess at all. But that could be one reason.
Great. Then I guess we now go on with the next talk, and then maybe at the end we have further questions for all the speakers. Thanks so much, both of you again. So I will stop.